Okay, as promised, we're going to get back to the second half of chapter 4. We're going to talk a little bit more about uh, some actual science of consciousness and less about the philosophy of consciousness and experience and awareness and qualia and that sort of stuff. One way to think about this is to think about ways in which our actual consciousness itself can vary in terms of what are known as states of consciousness. One of the things that we're not going to cover in detail in this set of notes are the so-called altered states of consciousness, although there's a little bit more information in the book about these kinds of things. But under that heading would be examples such as hallucinations. So when we talk about having perceptual experiences and qualia, those are experiences that are triggered by sensory inputs. Hallucinations, on the other hand, are similar sorts of perceptual events or qualia that are not actually caused by sensory inputs, but are presumably caused by other states originating within the brain itself. However, the exact nature of hallucinations is one of those areas in psychology that is also actually quite difficult to understand. Certainly we can study the conditions under which hallucinations occur, whether they are drug-induced states, as also listed here, or whether we're talking about something like some sort of psychological disorder, such as bipolar disorder or schizophrenia, that might cause these kinds of situations. But the actual nature of a hallucination itself, and we can certainly do something like fMRI research to understand brain areas that are involved, though it remains difficult, because in order to do that, what you want to be able to do is you want to have a person in the fMRI machine while you're scanning their brain experiencing hallucinations. And people with schizophrenia, for example, don't necessarily experience hallucinations constantly. So you have to make sure that you have them in the MRI tube at that exact moment when they are having a hallucination. And you have to rely on them to self-report that they are experiencing the hallucination at a given time. And that, of course, then takes us into other methods that we have seen in chapter 1 that are highly problematic, like introspection, that we have to let the person tell us that they are hallucinating and trust that that is, in fact, a correct response and truthful response. And then we might be able to see. And of course, we could induce hallucinogenic drugs in a person in an fMRI tube, though I'm not quite sure that that has really been done, primarily because it is currently not a part of US federal law to allow that kind of research with hallucinogenic drugs. Nor does it sound altogether pleasant to be under the influence of a hallucinogenic drug while inside a closed MRI tube, but that's another issue entirely. And in the middle there, we have the affective disorder. So here the idea is, I mentioned bipolar disorder, but we might claim that so affective disorder refers to any disorder of mood, right? So major depression is an affective disorder. Bipolar, or so-called manic depression, is another affective disorder, and even anxiety disorders could be considered affective disorders. And these certainly are ways in which our mental states can also be altered. And we'll have a lot more to talk about with these things when we get to the relevant chapter near the end of the book. But we're going to talk here instead about waking and sleeping, primarily sleeping. So let's first talk about this. We sleep and sleep itself represents a rhythmic behavior, or what we'll also call a periodic behavior. Periodic refers to the idea of a behavior that is cyclical in nature or repetitive. And when we talk about periodic cyclical behaviors, we talk about things that occur on specific time periods, right? That's what periodic means, is that there is a consistent periodicity or time interval associated with the repetition of the behavior. In fact, there are lots of different kinds of periodic behaviors that we see in animals of all kinds, as well as in humans. So we can define different kinds of periodic rhythmic behaviors. There are the so-called circannual rhythms, right? Circannual here referring to periodic behaviors that occur on a yearly cycle. So for example, bird migration. Birds that are flying south for the winter and back north for the summer is a circannual rhythm. It's a behavior, and it occurs periodically, right? Infradian rhythms occur on cycles that are less than a year, such as the monthly human menstrual cycle, or in the case of other mammals, the estrus cycle. And then there are those that occur on a daily, so of course that counts under infradian, but now it's more on a daily cycle. And that's where sleep comes in. Sleep is something that humans and most animal species do on a circadian basis, meaning daily. And then there are cycles that occur several times within a day, and therefore operating on a shorter time cycle. And those would be called ultradian rhythms. Eating, for example, is something that we do typically 3 times a day, although, of course, that may be less of a biological component and more of a social component. But generally, the rule is that we get hungry and eat multiple times throughout a given day, and that hunger cycle is something that occurs on an ultradian period, something less than a day. So we're suggesting that these cyclical behaviors are in fact biological in nature. And we need to understand the biological mechanisms of how these kinds of systems work. What determines the rhythm setting, right? How is it that something happens on a yearly basis or a monthly basis or even a daily basis, as in the case of the circadian rhythms? Well, these 3 terms relate to periodic behaviors. First is the Zeitgeber. That's a German word that means time giver, right? And what it really just means is that there has to be something in the environment that regulates the timing of the cycle. So in the case of circadian sleeping cycles, that's a relatively straightforward process. It is the daylight and nighttime, right? It's the sunrise and sunset. And the timing of those, which changes throughout the year, regulates the timing of our biological rhythm for sleeping and waking. And that would be the circadian rhythm. For birds that are migrating, there are other seasonal temperature changes that would be the Zeitgeber for their circannual rhythms. These Zeitgebers, I have referred to it as an environmental event, and that is true in the case of those 2 examples, but that may not always be exactly the case because, for example, we talked about the example of eating, and in this case, the Zeitgeber is actually fluctuations in our own body's energy levels. Blood sugar levels primarily fluctuate throughout the course of the day, and as they drop, that triggers hormonal processes that trigger our feelings of hunger and therefore eating behaviors. Same is true of the menstrual and estrus cycles. Those are triggered by internal hormonal processes. But keeping on with something like the sleep-wake cycle as a circadian rhythm, the Zeitgeber being sunrise and sunset, and the timing of that as it varies throughout the year, the effect that it has on us is a process called entrainment. So the idea here is that the cycle of sunrise, sunset occurs on its own, but as it varies throughout the year, it entrains our internal biorhythms. So to think about what that word means, to entrain something, just think about the idea of an actual train, in which case all of the various cars of the train are attached to each other, and therefore their motions become linked to the engine that drives the train. And so in this case, the sunrise and sunset becomes the engine that drives the train, and we are the cars of the train, right? But as it also turns out, there is an internal system that gets entrained by the Zeitgeber, but that internal system itself becomes kind of an internal controller that regulates all of the various parts of the systems inside of us that function as a result, and that's called our internal pacemaker. So again, going back to the sleep-wake cycle, the idea is that the environmental event would be sunrise, sunset. It entrains our internal pacemaker, which is in fact a neural mechanism called the suprachiasmatic nucleus, and I will explain that in here in a second. And in turn, the suprachiasmatic nucleus regulates the production of a variety of hormones and other neurotransmitters involved in our sleeping and waking behaviors, most crucial of which is a hormone called melatonin, which you can also take as a supplement to help regulate your sleep cycle. Well, there is that part of the brain that, as I just suggested, is our internal pacemaker for our sleep-wake cycle, the suprachiasmatic nucleus, or SCN for short. Now, what does that even mean? Well, nucleus here refers not to the nucleus of a single cell, which is maybe what you would normally think of when you see that word, but a nucleus in neuroscience anatomy, it refers to a collection of neurons working together as a group in the brain. In the peripheral nervous system, such a collection of neurons working as a functional whole is called a ganglion, but in the brain, it is called a nucleus. And so the suprachiasmatic nucleus is a cluster of neurons in the brain that sit just above the optic chiasm. And you don't need to know exactly what that is. We may talk about it a little bit in chapter 5 when we talk about the sensation and perception. The optic chiasm is an area of the brain where the optic nerve from both eyes conjoin with each other, right? So you have two eyes and an optic nerve that leaves each eye. And as those two nerves head back to the brain, they converge and cross over each other, and they form something like an X shape in front of the brain. And that's called the optic chiasm, is that X-shaped conjunction of the two optic nerves. Just above that, and that's what the prefix supra means, is then found the suprachiasmatic nucleus. Now, it is relevant to talk about the optic nerves here, because how is it that the suprachiasmatic nucleus is entrained by daylight, is entrained by variations in sunrise and sunset? Well, it has to be a visual signal, right? How else would a set of neurons inside the brain know when the sun is coming up, and therefore when it's light outside, or when the sun is going down and it's getting dark outside? It has to be visual in nature. So we have a special pathway for that. In addition to the optic nerves that send signals onto the brain that are used for conscious visual perception, again in chapter 5 content, there is another pathway coming from the eyes, from what are called the photoreceptors in the retina, which is the layer of cells inside of your eyes that contains the light-sensitive neurons called photoreceptors. And so this signal coming from the photoreceptors in the retina to the suprachiasmatic nucleus, which also should mention is actually part of the hypothalamus, which we learned about in chapter 3, which regulates a lot of our motivational and hormonal driven behaviors, is called the retinal hypothalamic pathway. And this is the signal that allows light to activate some receptors, which then sends a signal to the hypothalamus, specifically the SCN portion of the hypothalamus. And in this case, this is a visual input that has nothing at all to do with conscious visual perception, but instead is an unconscious process that, excuse me, that regulates our sleep-wake cycles, because the activity of the SCN will now become entrained but by the daylight cycles, and it becomes rhythmic in nature. There are other pacemakers that are involved in this process, some in the retina, some in a part of the brain called the pineal gland, but primarily we're going to focus on the SCN. And what's important about the SCN and its rhythmic activity here is that as an internal pacemaker, it can function on its own for some period of time. So what that means, for example, is that if you took a person and you locked them in a room with a single light on that was on 24 hours a day, this person would still get sleepy every now and then. And that is a result of the rhythmic activity of the SCN and its ability to control other parts of the brain, including the pineal gland, which is where melatonin is primarily produced. But in this case, what you would find is that in the absence of its connection to information about sunrise and sunset, if we kept this person in this kind of locked room without any knowledge and ability to see this daylight and nighttime, that eventually the SCN will become dissociated. It will drift its timing away from the Zeitgeber of sunrise and sunset. So what we see here is that entrainment is lost, right? So the rhythmic activity of the SCN occurs regardless of any entrainment with an external stimulus. But in order to keep our sleep-wake cycle entrained correctly to daylight, we need that visual input. Some things we know about this here is that we know the importance of the SCN because if we damage it, we've talked about doing that kind of research back in chapter 3, lesioning parts of the brain, such as lesioning the amygdala to see that it causes a loss of fear in animals. But in this case, lesioning the suprachiasmatic nucleus causes a loss of the periodicity in these kinds of behaviors, right? So what that means is that even though they still do it repetitively, especially sleep, but all these other behaviors too, because all of these things are regulated through the hypothalamus, but it becomes kind of haphazard and random, right? Meaning that there's no actual rhythm anymore, right? It just occurs haphazardly. The rhythmic aspect of it, the repetitive cycle, is lost. We, of course, can measure activity of the suprachiasmatic nucleus. That is, we can just record using electrodes the activity of the neurons in the suprachiasmatic nucleus. And what we find is that they are more active. They fire at a higher rate during the daytime light period of the day-night cycle, and then they fire less during the darker period of the cycle. And as I had mentioned, even if we were to take a person and put them into this room to cut them off from the external daylight information, the neurons of the SCN still fire in a rhythmic way. They just lose their entrainment to the world. Now, another way in which this has been done is rather than keeping a person locked in a room, what we can also do is cut the nerves that feed into the SCN from the eyes. Now, we're not going to do that with a person, but that has been done with animals. And what we'll see is that the SCN is still rhythmic even without that visual input. And even if you also cut away the output as well. So you can set it so that the SN is not receiving any input and is not sending any output to any parts of the brain. And the neurons are still firing rhythmically, even though that rhythm will eventually drift away from any actual daylight cycle. So that's what causes us to sleep. We can also then measure what happens when people sleep. And so we talked a little bit in the previous chapter about using electroencephalogram, or EEG, which records the repetitive rhythmic brain activity. And so when we speak of rhythmic brain activity, we should hopefully you're making the connection here that, of course, that could include the neurons of the SCN, but other neurons in the brain are also firing rhythmically under very rapid repetitive behaviors here. And in this case, we talk about repetitions, rhythms that are occurring several times per second. So an even faster kind of biological rhythm is what we're going to see with various brainwave measures. What also might be useful to do is to measure muscle contraction, and that uses a technique called electromyogram, or EMG. And what this does is it records the electrical activity of the motor neurons that innervate the muscles and cause muscles to contract. We can do that by measuring facial muscles. Of course, we can do that for the whole body, but for sleeping, it's oftentimes done by measuring muscle contractions in the face. And we can use EOG, electrooculogram, which really is a form of EMG, because what it does is it records your eye movements, and eye movements are controlled by muscles that surround the eyeballs themselves, the orbits of the eye. And so the electrical innervation and contraction of those muscles are detected by sensors mounted typically on the temples, and also one between the eyes, right between the eyes on the forehead. So let's talk about what we learn primarily from using EEG to look at brain waves. Well, there are different kinds of rhythms, and one simple breakdown is to talk about, at least when we are awake, alpha rhythms and beta rhythms. So alpha rhythms are those that occur in the vicinity of 8 to 12 hertz. So what does that mean, 8 to 12 hertz? This means that they occur 8 times per second up to 12 times per second. That's what hertz means, is how many cycles per second, how many repetitions per second. And by repetitions, I mean, when I'm talking about a brainwave, I'm talking about a pool of neurons that are all firing their action potentials in synchrony, right, as a group. And this firing of action potentials in this population or group of neurons occurring repetitively 8 times per second or 12 times a second. So that means you're looking at a bunch of neurons that are all working together, firing action potentials in synchrony 8 to 12 times per second. These alpha rhythms are associated, I should say, when people are awake, but are in a fairly restful, calm state. Oftentimes when people are asked to do things such as meditate and relax, you will see alpha rhythms occurring. In contrast to that, when someone is more alert and active, you start to see faster brain waves going upwards from 13 to 30 hertz, and there's less synchronization. So now you start to see more chaotic patterns of brainwave activity where the neurons are less synchronized, and some neurons are firing at 30 hertz and other neurons at 15 hertz, for example. So you see that different groups of neurons are now doing different things and they're firing at different rates. Whereas with alpha rhythms, you kind of see this synchrony across larger brain areas. You start to see desynchrony as brain areas start to do different things when people are more alert and active. And that might make some sense, right? Because when we're alert and active, oftentimes that means we might be doing some multitasking kinds of things. Whereas when you're sitting still quietly, meditating or just relaxing, your brain can synchronize itself. Now, when we're sleeping, on the other hand, we're going to see that there are other kinds of brainwave activities. So we can talk about first two ways of talking about sleep. One is non-REM sleep, and then REM sleep. REM sleep is referred to as rapid eye movements sleep, because that's what we can detect with the EOG or EMG, is the rapid movement of someone's eyes during sleep that occurs during special stages called REM sleep. But there are many other non-REM stages of sleep as well that are associated with slower brain waves compared to the alpha and beta rhythms. Though in contrast, REM sleep oftentimes is associated with some fairly fast brainwave patterns that are referred to as theta waves, as well as some beta waves. So we can look at some of these in this picture. So these graphs here show the wave-like patterns that can be detected with EEG, right? Each spike in this wave would correspond to a pool of neurons all firing their action potential at once, and that creates a positive spike in the waveform. And then they quiet down, and that causes the dip in the waveform, and then they all fire again, and that causes a spike. And so these repetitive spikes and dips reveal the repetitive firing of neurons. So here we see, for example, on the top of this chart, typical beta waves of a person who is awake and alert, right? So 13 to 30 hertz firing. As someone starts to fall asleep, the first thing that you do is you lay down and you relax and close your eyes. And so we would expect the brain to show some slowing and synchronization. And that's what the alpha waves look like, just below. And then we fall asleep. So one thing that might happen initially is that we start to show some theta waves, right? So these are slower even, right? So theta waves are happening in stage 1. They range from 3.5 to 7.5 hertz. So that is 3.5 to 7.5 times per second are the wave patterns. Now, where do you see that in this graph here? Because one of the things you might notice, for example, is that as the waves slow down, what that's supposed to look like is it's supposed to look like increased spacing between the peaks and the valleys of these waveforms. And that's really obvious going from beta waves to alpha waves, right? But is that necessarily obvious when we look at this right here? Well, what you're seeing here is a new pattern occurring. So you still have a lot of these really densely packed wiggles, which are really the alpha waves that we're seeing above. But notice that there's something else superimposing onto it. There's a tendency for this to go up and down, right? There's another pattern emerging on top of the alpha waves, and that's the theta wave. And the theta wave is approximately half the speed of the alpha wave. But then we enter stage 2, and what we see in stage 2 are what are called sleep spindles. And as it's described here in the text, short bursts of increased EEG activity. That's what we're seeing right here. So we start to see, you can see the theta waves, right? The slower up and downs of the entire graph. But embedded into that are these rapid little bursts, right? These peaks and valleys of the waveform occurring very close together in time, rapidly then, at these kind of haphazard rates. And those are sleep spindles, right? Occasionally, there are also these high amplitude spikes, and those are called K complexes. And that's not necessarily a great example of a K complex in this particular diagram. And then in stage 3, what we're going to start to see are even slower wave patterns, and these are called delta waves. And so we can see here these larger up-down patterns. Those are these slower waves. You can still see some of the faster alpha and theta built into there, but now, of course, even the slower, larger patterns are called delta waves. Finally, even get to stage 4, we start to see more regularity in all of this, and we start to see that it is primarily just delta waves that are present in stage 4. So what we've seen here is really a transition, right? Going from beta waves when we're alert that are very fast to alpha waves that are more synchronized and slower. And then in the first earliest stages of sleep, we're starting to see some slower phases start to appear, but they're irregular as we move through stages 1, 2, and 3. And then eventually it kind of settles into a more rhythmic, consistent delta wave pattern. And that's one way to think about our stages of sleep, is to get into that stage 4 deep sleep. We're going through some of these more irregular brainwave patterns before we get into that nice deep delta wave sleep. So what we have just described are the stages of what would be considered non-REM or NREM sleep, right? There are other things happening during NREM sleep. For example, our body temperature will gradually decline. You may find yourself feeling somewhat warm in bed when you first go to bed, but at some point in the middle of the night, you might happen to wake up and find yourself that you're freezing, right? And that's because during NREM sleep, your body temperature decreases. The hypothalamus will also start to trigger the pituitary to produce growth hormone, right? Which certainly, of course, is true for childhood when we are still growing. But growth hormone may play a role in the body's healing process as well. And so there's going to be some healing that takes place if there's a need for that, of course. We can also dream. A lot of times people think, oh, we only dream during the so-called REM sleep, but that's not true. We can dream during NREM sleep, though when we ask people to report their dreams, and one way in which this is done is that we wake people during their sleep. Right. And so if we wake them during NREM sleep, we can ask people, were you dreaming just now? And that's one way in which we know that people are dreaming, because we can't read their minds, right? You can't necessarily look at a brain scanner and know for sure that someone is experiencing a dream, whether it's MRI or EEG. But if you wake them and ask them if they were dreaming at that point, they can tell you, right? But the difference is that how vivid that dream was and how easy they can remember it is less the case during NREM sleep. But if you wake people during REM sleep, they will report that they were having more vivid dreams and be able to describe them in more detail. Night terrors, which are these kind of sudden waking events, where people just wake up suddenly feeling afraid, maybe even screaming, and it's commonly observed in early childhood, often occur during NREM sleep as children are moving through those first 3 stages of sleep before they get into deep sleep. Something interesting about REM sleep, besides the vivid dreaming, is that even though the eyes are moving, the rest of the body oftentimes tends to be paralyzed, and that's called atonia, where the motor neurons of the body are now inhibited and are unable to fire action potentials, and therefore it becomes difficult or impossible for us to voluntarily move our bodies. Sometimes people report this idea of sleep paralysis. If you happen to be conscious for some reason during REM, sometimes people report this with something like lucid dreaming, that they feel paralyzed and they can't move. And sometimes it's described as frightening as a result of that. Our next question is the why. Why do we sleep? What is the function of sleep? Here we're going to talk about, we could talk about the whole, growth hormone and healing and growth issues, which certainly are an important thing in childhood and play a big role in while children sleep a lot. Babies sleep a lot, right? And that's because they're growing a lot. In adolescence, we sleep a lot as well. And of course, we grow a lot during adolescence as well. And as people get older, they sleep less. And that may have something to do with growth issues not occurring quite as much as we get older. But there are other things, of course, and other reasons that we are sleeping. So in this case, we know that sleep plays a role in memory formation. And therefore, there may be something going on with these brainwave patterns that help kind of encode and organize memories of events that happened during the previous day. One of the ways that we know that is that if people are deprived of sleep, they exhibit memory problems. They start having trouble remembering things, and they also show impaired cognitive performance. So if you give people tasks to measure their short-term or working memory abilities or their attention multitasking skills, they perform very poorly if they are sleep deprived, as well as just not being able to remember things. And of course, we can do that with humans, where we just ask them that. But we can also do learning experiments. So if you train animals to learn various things, but you deprive them of sleep, we see that interferes with their learning. One example of this here, especially this kind of interesting research, there's a part of the brain, the hippocampus, we learned about it in chapter 3, that plays a role in encoding long-term memories. But it also seems to be that the hippocampus is involved in what we call place learning and place knowledge, right? So spatial learning and knowledge. And there are specific cells called place cells in the hippocampus that, that fires in response to certain places. So in the case of rat research, for example, the famous thing we might do with rats is learning a maze. And what we can see is that at certain junctions or locations in a maze, there would be different kinds of hippocampal place neurons that fire when the rat is in those particular locations. So one of the things that we oftentimes see doing here is that when training the rat to engage in a food search task in a complex environment such as a maze, we will find certain place cells are active as they perform that task. And then what we'll also find is that later when the rats are sleeping, those same place neurons are active during their sleep period, suggesting that this kind of repetitive activity of the hippocampal place neurons is playing a role in embedding and encoding those memories for the rats so that they learn that later. Because learning things such as where in my environment is the source of food is an important thing to be able to remember, perhaps. And sleep plays a role in that process. Now, after we're done sleeping, we need to wake up. And we mentioned this in chapter 3, that this is the function of the reticular activating system of the midbrain. It's to help keep us asleep when we're asleep. But when it's time to wake up, its job is to wake us up. So it is a reticulum, which means that it's a collection of both cell bodies or nuclei, as well as nerve fibers, which refers to the axons. Right, so it contains cell bodies, but it also contains axons that pass through the midbrain from the brainstem, and from the brainstem is coming a signal from the spine, which is bringing sensory signals in from the body, right? And what this can do is it can either block those signals to keep the brain asleep by preventing sensory signals from coming in, or it can allow those signals through, arousing the brain and therefore allowing us to wake up. So certainly it plays a role in our sleep-wake behavior, but it can also arouse us to behavior as well. So even during times when we're awake, our arousal levels throughout the day can change, and that could be a function primarily of the reticular activating system, sometimes making us feel more alert. Think about the activity of the sympathetic nervous system, for example, which we've talked about before. And one of its jobs is to increase our alertness during periods of danger, right? That's the fight or flight response. And that kind of arousal coming from the peripheral sympathetic nervous system eventually has to arouse the brain, and that is also working through mechanisms in the reticular system. One of the things that we can do, again, just to how do we know these things? One of the ways in which we know these things is to apply an EEG cap to someone to record their brainwave activities. And if we stimulate the reticular system with electrical signals, we start to see things like beta waves, right? The kinds of waves that are associated with being awake. If we lesion the reticular system, it produces very slow wave EEG, right? Which is similar to deep sleep. But of course, particular lesions to the reticular system make it impossible for the brain to be aroused, and therefore the patient will be lapse into a coma. Obviously, we don't do that kind of experimental research with humans, but with animals that can be induced. But what we also do know is that in some cases, it is possible to have damage to the reticular system, perhaps due to a tumor or other neurodegenerative conditions, which leads to slow wave EEGs and coma as well. And again, just a reminder, we had pictures like this from chapter 3, but we can see how sensory information arises up through the spine and into the brainstem, passing through the reticular activating system, which can then make it on to the hypothalamus, thalamus, and routed to other areas of the brain to wake up those parts of the brain, or in the case of sleep, to prevent that signal from being allowed to enter into the brain and help keep us asleep. Well, that's the short and sweet of brainwave activity and sleep-wake cycles. And that's the end of chapter 4.